Welcome back, class. This is Silvana Iconi with Lecture 5, the viruses. So in this lecture we're going to focus on the broader aspects of virology, that is the study of viruses. We'll look at a brief history of the discovery of viruses, their general properties and the structure of viruses, the ways in which they're cultured, and very briefly, viral taxonomy. Now we're going to take a look at the general properties of viruses. So a complete virus particle is referred to as a virion, and a virion consists of one or more molecules of DNA or RNA enclosed in a coat of protein, although some viruses have additional layers, which we'll talk about in just a moment. Now, viruses can exist in two phases, the extracellular and the intracellular phase. Virions are the extracellular phase. They possess very few, if any, enzymes, and they cannot reproduce independently of living cells. In the intracellular phase, viruses exist primarily as replicating nucleic acids that induce the infected host cell metabolism to synthesize additional virion components, eventually synthesizing complete virion particles that are then released from the cell. So in summary, viruses are different from living cells in at least three ways. One, their simple acellular organisation. Two, the presence of either DNA or RNA as their nucleic acid, but not normally both. And three, their inability to reproduce independently of cells and carry out cell division as prokaryotes and eukaryotes do. Here we can see the generalized structure of viruses. So almost all viruses fall into one of these two categories. On the left we have what's called a naked virus, and to the right we have the enveloped virus. So the simplest viral structure is that of the naked virus. It consists of a piece of nucleic acid, which you can see indicated in red, and surrounding this is a protein shell called a capsid. And together, the nucleic acid and the capsid together are referred to as a nucleocapsid. In B, we have the more complex type of virus called the enveloped virus, and it still consists of the nucleocapsid, but to the outside of the nucleocapsid we have a flexible membrane called an envelope. And the envelope usually has viral proteins inserted into it, and these are called spikes. Virions range in size from about 10 to 400 nanometers in diameter. The smallest viruses are a little larger than cell ribosomes, whereas the pox viruses are about the same size as the smallest bacteria and can be seen in the light microscope. Most viruses, however, are too small to be visible just using a standard light microscope and have to be viewed using a scanning or transmission electron microscope. So now let's just summarise what we've learned about the structure of viruses so far. All virions contain a nucleocapsid, which is composed, or rather which contains one or more pieces of nucleic acid, either DNA or RNA. The nucleocapsid is surrounded by a protein coat called a capsid. Some viruses consist only of a nucleocapsid, others have an additional component, such as that external membrane called an envelope. Virions that are surrounded by envelopes are called enveloped viruses. Those that lack envelopes are simply referred to as naked viruses, as we saw on the previous slide. Okay, here we can see the tremendous variation in both size and morphology of different viruses. In A, we have one of the large pox viruses. This one's called the Yersinia virus. In B, paramyxovirus, which causes mumps. In C, the herpes virus. D, the Orff virus. In E, we have this characteristic bullet-shaped virus, rhabdovirus, and the rabies virus belongs to this group of viruses. In F and G, we have two examples of phages, that is, viruses that infect bacteria. In H, we have the adenovirus. Very familiar virus to everyone because it's mostly responsible for causing the common cold. In I, we have the influenza virus, of course, which is responsible for causing the flu. Then we've got some smaller viruses in J, K, and L, polyomavirus, picornavirus, and the X174 phage. And then at the bottom right of screen in M, we have the tubulovirus. Let's take a closer look now at the virus capsid. So a capsid is a large macromolecular structure which serves as the protein coat of the virus, as you can see in this figure here. The capsid protects the virus genetic material and also aids in its transfer between host cells. Capsids self-assemble from many copies of one or a few types of proteins, and the proteins that are used to build the capsid are called protomers. We've seen various morphological types of viruses, and this variation in morphology primarily results from the combination of a particular type of capsid symmetry with the presence or absence of an external envelope. There are three types of capsid symmetry: helical, icosahedral, and complex. Helical capsids are shown here. They're shaped like hollow tubes with protein walls, and the tobacco mosaic virus provides the best studied example of a helical capsid structure. In this virus, the self-assembly of protomers into a helical or spiral arrangement produces this long, rigid tube, about 15 nanometers in diameter by 300 nanometers long. And this type of capsid encloses an RNA genome, which is wound in a spiral and lies within a groove formed by those protein subunits. But not all helical capsids are as rigid as the tobacco mosaic virus capsid that we just saw on the previous slide. Here we have the influenza virus, and the influenza virus genome is enclosed in thin, flexible helical capsids that are actually folded within an envelope. In A, we have a schematic view of the influenza virus, and these viruses have what are called segmented genomes consisting of 7 to 8 different RNA molecules. In B, we have an electron micrograph image of the influenza virus particles. This is at 350,000 times magnification. The second type of symmetry commonly seen is the icosahedron. The icosahedron is a regular polyhedron with 20 equilateral triangular faces and 12 vertices. It's one of nature's favourite shapes, and the icosahedral capsid is the most efficient way to enclose a space. The icosahedron is a complex structure. The capsids are constructed from ring or knob-shaped units called capsomers, Each capsomer is usually made of 5 or 6 protomers. Pentamers have 5 subunits, hexamers possess 6 subunit capsomers. Here we have some examples of icosahedral capsids. Starting with A, this is the canine parvovirus, which contains 12 capsomers. Across to B, this is a computer image of the polyomavirus. This is a virus that causes a rare demyelinating disease of the central nervous system, and it contains 72 capsomers. In C, we have the adenovirus, which consists of 252 capsomers, and this is at 171,000 times magnification. And in D, we also have the adenovirus, but this is a computer simulated model. So interesting to note that self-assembly of capsids is a remarkable process that's really not fully understood at this time. Now, although most viruses have either the icosahedral or the helical capsids, many viruses don't fit into either category. And this is the third type of capsid symmetry called complex symmetry. The pox viruses and the large bacteriophages are two important examples. Here we have some examples of viruses with complex symmetry. So in A, B and C upper panel, we have an example of a pox virus called the Yersinia virus. It has an ovoid structure, as you can see. The nucleic acid is associated with proteins and surrounded by an internal core membrane, as indicated. It's also surrounded by an outer membrane covered with fibres. In the lower panel, in both A and B, we have an example of a large bacteriophage, which is even more complex in structure. It has what's called binal symmetry. That is, it has an icosahedral head, and you can see the capsid head indicated in A. The tail is helical in structure, and the tail is connected to 6 tail fibres. Let's turn our attention now to the viral envelope. Many of the animal viruses and plant viruses, and at least some bacterial viruses, are surrounded by an outer flexible membranous layer called the envelope. Animal virus envelopes usually arise from the host cell nuclear membrane or the plasma membrane, and the lipids and carbohydrates within the membrane are normal host cell constituents. But in contrast, the proteins found within the envelope are encoded for by virus genes and may even project from the surface of the envelope as spikes, otherwise known as peplomeres. Here we have some examples of enveloped viruses. In A, we have a schematic of the influenza virus, a very well studied example of an enveloped virus. The spikes project about 10 nanometers from the surface of the envelope at 7 to 8 nanometer intervals. Some of these spikes possess the enzyme called neuraminidase, which functions in the release of the mature virions from the infected host cell. Other spikes contain what are called hemagglutinin proteins, and these are so named because they can bind the virions to red blood cell membranes and cause the red blood cells to clump together, otherwise known as agglutination. This is called hema glutination. Hemagglutinins participate in virion attachment to host cells. In B, we have another example. This is the rabies virus with that characteristic bullet shape. And you can see the spikes projecting from the surface of the envelope. In C, we have the HIV virus or human immunodeficiency virus. In D, a herpes virus. And in E, the Semliki forest virus. This is a virus that occasionally causes encephalitis in humans. So we've looked at the virion outer structures. Now let's take a look at what's normally found inside the virion particle. It was originally thought that all virions lacked enzymes. However, as we've just seen in the discussion of the influenza virus, this is not the case. In some cases, enzymes are associated with the envelope or capsid, and we've just seen an example of the neuraminidase enzyme that's part of the spikes embedded in the envelope. But most viral enzymes are located within the capsid. Many of these enzymes involved in replication of the virus nucleic acid. For example, the influenza virus uses RNA as its genetic material, and it carries an enzyme that can synthesize RNA using an RNA template. These enzymes are called RNA-dependent RNA polymerases. So although viruses actually lack true metabolism and they cannot reproduce independently of living cells, they may carry one or more of the enzymes that are essential to the completion of their life cycles. Complete virion particles also contain some form of nucleic acid. And virions are incredibly flexible and unique with respect to the nature of their genome. They employ all four possible nucleic acid types. They may contain a single-stranded piece of DNA or a double-stranded piece of DNA. They may contain single-stranded or double-stranded RNA. The size of the viral genetic material also varies greatly. So the smallest genome is around 4,000 nucleotides, which is just large enough to code for 3 or 4 proteins. At the other extreme, some of the large bacteriophages, herpes virus and the Yersinia virus have genomes spanning 1 to 2 by 10 to the 5 nucleotides. And this enables them to synthesize over 100 proteins. Some of the genomes can be either linear, others are circular, and in some viruses, the virus can switch from one form to the other. Another important characteristic of viral nucleic acids, particularly the DNA viruses, is that their genomes often contain unusual nucleotides. So for example, the T-even phage of E. coli have 5-hydroxymethyl-cytosine instead of cytosine. And the hydroxymethyl group is often modified by attachment of a glucose residue. The different types of viral nucleic acids and virus examples are summarised in this table. So these differences that we've seen in virus structure and viral genomes have important implications for the mechanism that a virus uses to reproduce within its host cell, that is within the cell that it's infecting. And even among viruses with similar structures and similar genomes, each one can actually exhibit very unique life cycles. However, here we're going to look at a general pattern of virus reproduction. And you can see that there are 5 steps here to the right of screen. Because a virus needs a host cell in which to reproduce, the first step in the life cycle of a virus is attachment to a host cell. This is followed in the second step by entry of either the nucleocapsid or just the viral nucleic acid into the host cell. And we can see here in the second step that only the nucleic acid itself is entering the host cell. If the nucleocapsid enters, then uncoating of the genome usually occurs, that is, release of the nucleic acid from the nucleocapsid. This usually occurs before further steps can occur. So once free in the cytoplasm of the host cell, genes encoded by the viral genome are expressed. That is, the viral genes within that nucleic acid are transcribed and translated. This allows the virus to take control of the host cell's biosynthetic machinery so that new virion particles can be made. The viral genome is then replicated and viral proteins are synthesized. New virions are then constructed by self-assembly of coat proteins with the nucleic acids. And then finally, in the last step, the mature virions are released from the host cell. And of course, virus reproduction can vary dramatically. So, for example, some viruses are released by lysing the host cell, which is what's happened here, whereas other virion particles simply bud from the host cell without lysing the cell.